Okay, so let me introduce Dr. Ramesh. Okay, now, Dr. Ramesh is currently working as a senior lecturer under Faculty of Science and Marine Environment, University Malaysia Terengganu since 2018. So he had experience as a postdoctoral researcher in Tianjin University, China for a year, and he was also a clinical research coordinator in ACER in India for 2 years. So currently, he has filed one patent, one copyright, published a book chapter, and have has he owns 37 research articles in high reputed index journals. Okay, so without further ado, let's welcome Dr. Ramesh.
Thank you, Dr. Kodak. Am I audible?
Yes, you are audible. You can start your
Thank you.
session.
First of all, I would like to thank you for giving me this wonderful opportunity for this. To okay. Uh, so, good afternoon to one and all present here. Let me share my slides first. Can you all able to see the screen? Shall I start? So, without further delay, let me continue. So, I think Dr. Kodak already gave a brief introduction about me. So, I'm just skipping this slide. So, I'm Dr. Ramesh from India, currently working as a senior lecturer at UMT under Faculty of Science and Marine Environment. Today I'm going to share a few things about my research expertise, which is pharmacognosy. Okay, first, pharmacognosy. Pharmacognosy is the. Can anyone know what is pharmacognosy? Pharmacognosy is the study of natural products such as plants, animals, and microbes, which are used for the medicinal and health benefits, which include analysis of their biological, chemical, biochemical, and physical properties. It means in pharmacognosy, we will explore the natural sources such as plants, animals, and microorganisms to utilize their benefits for therapeutic applications. So, where we will explore their biological, chemical, and physiochemical properties. So, this word pharmacognosy is derived from two Greek words, which is pharmakon, which is known as drug, and gnosis is a knowledge. Okay, so it is a study of drug. We are going to gain the knowledge of the drug. Okay, it was first coined by the Austrian physician called J.S. Kivitt and then by a German scientist Sedler in 1850. This pharmacognosy is a vital tool for a drug discovery and development, and it plays a crucial role in the search for new treatments for a wide range of diseases. Actually, the use of natural products it starts from early ages, not only in the new say current era, because it starts from the early stages, from the history. Throughout the history, We are utilizing the natural product for medicinal applications, and many of the drugs which we are using today are also derived from natural products. According to the recent WHO report, almost like 80% of world population is still using the natural product for their primary healthcare needs. So what is natural products? Natural product is a chemical compound or substances which are produced by a biological source such as plants, animals, marine sources, and microorganisms that are found in nature are known as natural products. So these natural products are classified into two major classes. For example, primary metabolites and secondary metabolites. Here, the primary metabolites. So normally, any plant or animals or any microorganism which contains two kinds of metabolites, which is primary metabolites and secondary metabolites. In this, primary metabolites uh, will have an intrinsic function which is essential to the survival of the organisms which produce them. For example, carbohydrates, proteins, nucleic acids. So these things will be utilized by the own organism for their normal growth, development, and production. Whereas secondary metabolites, which have an extrinsic function that mainly affect other organisms. So mostly many secondary metabolites will have medicinal properties. Normally, in pharmacognosy field, researcher deals with the secondary metabolites found in the plants, animals, and microbial sources. For example, leaves, seeds of the plants, roots, stem, rhizophores, herbs, corals. In animals, it will be, they will go for the starfish, jellyfish, snake venom, sea cucumber. All these natural sources have secondary metabolites which can be utilized for the medicinal or therapeutic applications. So pharmacognosy, this pharmacognosy has significant cultural and historical importance. It has a wide range of written history all over the world. Pharmacognosy has been a key component in various systems of medicine, including Western medicine, Unani, Ayurveda, Orient, the Greek history, and the African system. So I'll just have a tell about a few things about what kind of where this Western medicine started, what they have contributed to the pharmacognosy. So first, this Western medicine originated from Mesopotamia and Egypt. This Mesopotamia is considered as the first origin of human civilization. The Sumerians, for example, the people of ancient Mesopotamia, developed uniform tablet of herbal medicines. Those tablets are still preserved in British Museum. In Egypt, the information had written on paper called Papyrus Ebers. It consisted of 800 prescriptions mentioning about 700 drugs. So the first named pharmacopoeia, named London Pharmacopoeia, was published in 1618, and then the British Pharmacopoeia was published in 1864. This pharmacopoeia is an official publication which contains a list of medicinal drugs with their effects and their direction of usage, how to use these drugs. So it has a proper publication, and these publications provide a useful information for the current drug discovery process. Next, the Unani. This herbal system was developed by Arabian Muslim Ibn Sina. He was a prince and ruler. He was a very brilliant pharmacist and physician who wrote a book called Kitab al-Shifa, which means Book of Healing. This book was written on Arabic language, and this is a great contribution of Ibn Sina on medicinal and pharmaceutical sciences. Next, coming to the Ayurveda, which is from Indian origin. Ayurveda is the term for traditional medicine of ancient India. The word Ayur means life and Veda means study, which is study of life. So these Ayurvedic writings were divided into three systems, which is Sharaka Samhita, Sushruta Samhita, Ashtanga Sanghara. The oldest writing was Sharaka Samhita, which is between 6th to 7th century before the Christ. Here, this book describes use of many metallic drugs such as iron, mercury, sulfur in combination with the herbs. So nowadays we are utilizing many nanoparticles using silver nanoparticle, gold nanoparticle, zinc nanoparticles. All these were already been utilized by our ancestors. It is like 6th to 7th century before the Christ. So all these informations were already written in the Ayurveda, the book called Sharaka Samhita. Next, the Orient. This is originated from Chinese, Japanese, and Tibet. The Orient herbalism was very old and called Kampu. These written documents were made by the king Shenyang and Shang. Shenyang investigated medicinal value of several herbs and written a book named Pensi Sau or Native Herb. The next is a Greek history. So all these histories, what to say, all these systems have a key component of pharmacognosy in it. Some of the early naturalists, scientists, and physicians who contributed enormously to the development of human knowledge about medicinal plants. For example, Hippocrates, who is called as a father of medicine. He was the first natural doctor who utilized simple remedies such as vinegar, honey, herbs, etc. in healing. He is also known to collect and identify a number of medicinal plants. Next, coming to Aristotle, he gave the philosophy of medicine. He listed more than 500 plants of medicinal importance. Then Theophrastus, he gave a scientific basis for the use of plant as a medicine. Then Galen, which is a Greek pharmacist physician, he developed the method of preparing and compounding medicines by mechanical means. He was the originator for the formula of the cold cream. Finally, there's an African system also supported the pharmacognosy system because they keep information in their groups or tribes. They transfer the information from one generation to another. And these regions are richest source of medicinal plants and they need to be explored for still they need to be explored for the new drug discovery. So I already mentioned like many plants have medicinal importance and the compounds are been still in use for medicinal purposes. So I'm just going to share few examples of medicinal plants and from that plants some compounds have been derived and these derived compounds are already been used as a anti-cancer drug or anti-malarial drug or it can be used for hypertension etc. So these are the few examples. For example, Kaolin, which is derived from the plant source called Amivisniga, which is now being used as a bronchodilator, and the Galligin, which is also derived from the plant named Galena officinalis, which is now currently being used as an anti-diabetic drug. The, I think you might have heard about metformin, which is already been used for the diabetic patients. So this is the source of this medicine is originated from the plants. So like that quinine, vincristine, paclitaxel, all these compounds are derived from the type of plants and it has been vital, it has played a vital role in medicinal applications such as anti-malarial, anti-cancer and painkiller. So these are all few examples of marine organisms in drug discovery. So here the marine organisms including sponges, algae, corals are a promising sources of natural products with therapeutic applications. For example, here the anti-cancer drug trabectedin, which has been derived from a sea squid, which is found in the Mediterranean Sea. Likewise, another compounds such as vidarabin, citarabin, ginconate, all these has been derived from the marine organisms, and these are used as an antiviral drug. It has been used as a painkiller and anti-cancer agent. Next, I already mentioned there are plants, animals, and microorganisms. These microorganisms have a secondary metabolites and many compounds which are being used as a medicinal drug. For example, the compounds such as penicillin, cephalosporin, cyclosporin, which are isolated from the various microorganisms like penicillium, cephalosporin, all these compounds have been used as an antibiotic. So marine organisms such as bacteria, fungi, and actinomycetes are rich source of natural products. Many antibiotics have been identified and derived from marine organisms, and other natural derived microorganisms include rapamycin, mavastatin, anthracylin. All these have a major role in treating the cholesterol, anti-tumor activities, and it also acts as an immunosuppressive agents. Apart from that, there are some role of animals also. Some compounds have been isolated from the animals such as snake, frog skin, and the venom of the snake have been used from this venom. They have isolated many compounds which are still being used as a painkiller, AC inhibitors, anti-diabetic drug, painkiller, anti-venom agent, all those things. So these are the few examples of drugs which have been isolated and derived from the plant material or microorganisms or animals which have been still used as a therapeutic drug to treat various diseases. So, what is the scope of pharmacognosy? So, here the scope of pharmacognosy includes isolation, identification, and characterization of bioactive compounds from natural sources such as plants, animals, and microorganisms. It also involves the study of the biological activities and mechanism of action of natural products and study of the safety and toxicity of the natural products and their potential interaction with other drugs. It involves study of the pharmacokinetics and pharmacodynamics of natural products, which include their absorption, distribution, metabolism, and excretion. These natural products, the compounds isolated from the natural sources, are served as a model to synthesize new types of drugs. And the few drugs which are isolated from the plants can be directly used for a therapeutic purpose. It also The scope of pharmacology involves biosynthetic pathway investigation, cultivation and collection of medicinal plants, preparation of herbal formulation, development of tissue culture plants. So all these categories falls under the scope of pharmacognosy. So this pharmacognosy field involves multidisciplinary approach where it involves subjects such as botany, chemistry, zoology, ethnobotany, biotechnology, microbiology, marine biology, herbal medicine, phytochemistry, pharmacology, clinical pharmacology. So it is like a multidisciplinary approach. It utilizes all these kinds of these different fields to produce a novel drug. Produce a new drug. So, what is natural product drug discovery? So, we are finding a drug from the natural source. So now recent progress in extraction, chromatography, hyphenated techniques, and biotechnological advancement has opened new avenues and lines for pharmacognosys to enhance their natural product research. Nowadays, natural products play an important role at the early stage of lead discovery. What is called lead? A lead is a chemical compound that shows promise as a treatment for a disease and which may lead to the development of a new drug. Okay, so how they are identifying a lead in this natural product drug discovery process? So it will be done by two different process, which is like traditional approach and modern approach. So but traditionally, what they will do means they will collect the raw material. Means they will go for sampling. They will collect the plants or plant source or animal source, and they will collect the samples. Sorry, I hear like there is my slides are not moving. Is it? Thank you. Sorry for the interruption. I think there was some technical error, so I'll continue. So initially, I have discussed about what is pharmacognosy and what are the types of what are the histories involved in the pharmacognosy, those things, how we are isolating the compounds from the natural source. So there are two types of approaches, which is overview, traditional approach and modern approach. In traditional approach, what they will do, this is the overview of traditional process, where they will collect the raw material from the natural sources such as plants, microorganisms, or animals, and then they will do an extraction process with the help of many extraction techniques. And from the extraction, they will further isolate the active fraction with the help of several separating process. For example, they will involve like liquid partitioning or any other fractionation system to identify the active fraction. So then this fraction will be tested for their biological properties, as well as they will also characterize the chemical components and physical properties of this active fraction. Then this further fraction will be subjected to separation process, which involves chromatographic techniques. So with the help of chromatographic techniques, they will purify the active components, and they will evaluate their biological properties as well as physical and chemical characterization. Then further, with the help of advanced chromatographic techniques, they will isolate it, which is like HPLC, RPHPLC, those things and all, reverse phase liquid chromatography, those things, they will isolate a pure compound, and this pure compound will be the lead compound, which will be used for further drug discovery process. Okay, so this is the traditional approach. Next slide. So these are all I already mentioned, like from the raw material, they will undergo extraction process. This extraction process involves different types of techniques such as maceration, Soxhlet extraction, microwave acid extraction, ultrasound acid extraction, accelerated solvent extraction, and supercritical fluid extraction. All these extraction process will enhance the speed up the process of extracting the compounds from the natural sources. Okay, next slide. So then I already mentioned, from with the help of analytical and chromatographic techniques, they will identify the chemical components present in the natural sources. So these are all the techniques. These are all the spectroscopic techniques like nuclear magnetic resonance, infrared spectroscopy, mass spectrometry, UV visible spectrophotometry, and chromatographic techniques, and tandem mass spectrometry techniques, which are involved in identifying the chemical components present in the identifying and isolating the chemical components present in the natural sources. Next. So then once they have isolated the active fraction or active components or active extract from the natural source, they will undergo the biological evaluation. So they will evaluate the biological properties of these secondary metabolites. So which they will do some in vitro and in vivo assays to confirm the biological properties. So initially for they will test in the cells in the lab environment, cells, tissues, or organs, and they will go with the animal studies, and they will utilize like various kind of animals like rabbit, mice, mouse, and they evaluate their biological properties such as cytotoxic, antibiotic, antioxidant, UV protection, antimicrobial, anti-inflammatory, anti-obesity, and all these disorders they will check, and then they will identify the specific biological properties of the isolated compound or isolated fraction. Next. So in modern drug discovery process, so far we have discussed what is how they are using the traditional process to identify a lead compound. But in modern drug discovery process, with the advancement of many techniques, now they are identifying the target, molecular target. For example, receptors, the genes, enzymes, and regulatory elements, which is responsible for a particular disease. So, for example, in our body, if there is any deficiency, the particular receptor will be the culprit, or else particular protein markers will be the culprit. So initially they will first identify the molecular targets, and the, then they will identify the components present in the plants with the help of chromatographic and extraction technique. With their both knowledge, for example, the target and the sample, they will go for they will go for this molecule silico docking process, and they will exploit the leads from the traditional medicine, and they will synthesize the compounds, and sometimes they will do the in silico screening and identify the natural products. So for our next slide. Next, so see here, first they will identify the disease. So on, they identify a protein which involved in the disease, that is a target, and then they will find a molecule which is effective against the target. Once they have identified these two things, then they will check the match whether this receptor can bind with the particular ligand and whether this can easily inhibit or activate the expression or inhibition of the particular process. And then once it gets successful, then they will go for a preclinical testing with the help of mice or any other rodents. Then they will go with the human clinical trials, and then finally once it get passed, then they will go for the approval from the regulatory agencies, and finally the drug will go on to the market. So the whole process will take like almost like 10 to 15 years, and the expenses will be like almost like 1 billion USD. So drug discovery process is not a simple thing. So it's like uh, with the help of many expensive techniques, many advanced techniques, it nowadays become very easy. Next. So so far I gave a brief introduction about pharmacognosy, but now I'm going to explain what I am doing in this field. Okay, how I'm supporting this field and how I'm utilizing the techniques which involved in this field of pharmacognosy. Okay, so my research target is on photoprotection, wound healing, inflammation, and oxidative stress. Photoprotection. What is photoprotection? Photoprotection is a biochemical process which helps the organisms or humans to cope up with the molecular damage induced by the UV light. This UV light normally comes from the sunlight. Okay, sunlight contains a wide range of electromagnetic radiations, which is UV, visible light, infrared light. Okay, so in this, our my main target is on UV light. Okay, this UV light is of three types, which is UVA, UVB, and UVC. These three types of radiations are classified based on their wavelength. Normally, the UV length nanometer is 200 to 400 nanometer. So, based on the type of nanometers, for example, from 280 to 320 nanometer, it is considered as a UVB light. From 320 to 400 nanometer, which is considered as a UVA light, and 200 to 280 nanometers, a UVC light. Normally, the UVC light is very dangerous, and it won't enter into the Earth's atmosphere because the ozone layer will completely absorb the UVC light, whereas UVA and UVB, these two radiations are the culprit, which is responsible for the photoaging, immunosuppression, melanoma cancer, sunburn, skin cancer, etc. So, my target is prevent this UV light with the help of natural sources such as plants and marine organisms. Okay, so this is my first target. I will explain like how I search for the plant and how I identify contribute to this photo protection with the help of natural sources. Next, my target is wound healing. We all know the wound healing consists of four stages. First one is wound healing process of four stages: hemostasis, inflammation, proliferation, and remodeling. In this, my target is like whether any of the plant sources will enhance the any of these stages of wound healing process. So I will collect the plant sample or natural products or natural fractions, and I will test it for their wound healing properties. Okay. My next field of interest is inflammation. Inflammation is the first defense mechanism which occurs in our body if there is any external stimuli or foreign organisms enter in our body. Okay. So normally this inflammation response will be occur if we have obesity, diabetes, or any cardiovascular disorder, or any neurological disorders, or anything else. So if I target this inflammation, anti-inflammatory properties or anything, then it will be the cure for many type of these disorders. Okay. Then my another target is oxidative stress. Oxidative stress is an imbalance between the free radicals and antioxidants. Normally, our body is capable to scavenge the free radicals which are stimulated in our body, so it will maintain a balance. But due to modern lifestyle and other environment culprits like UV radiation, air pollution, smoking, other microorganisms, there will be an imbalance in our antioxidant profile. Okay, so which further leads to cancer, cancer, or any other heart diseases or any other disorders in our body. So the first thing which we need to focus is we have to balance the free radicals and antioxidants in our body. In our body itself, there are many kinds of antioxidant enzymes, and non-enzymatic antioxidant compounds will be there, which will fight against these free radicals. But we need more to balance it. So I'm searching the natural sources to balance the antioxidant properties in our body. So these are all the four major portion which I am looking into it, looking for the medicinal application. Next slide. So these are all my previous projects which I have been done since 2017. So my first project is on photoprotective properties of Xanthosylum retsa, where I evaluated the chemical, biological, and product formulation studies. Here, I checked the photoprotective properties of the traditional medicinal plant Xanthosylum retsa. Next, biological activity of various solvent fraction of Inonotus obliquus. This Inonotus obliquus is a mushroom, which is called the king of mushroom. So this mushroom I have isolated and identified the medicinal properties against the oxidative stress in the liver cells. So here I check the biological properties of this fungi in the form of fungisome. Fungisome is like I synthesize it into a nanoparticle. I will explain this in the next slides. So then I have checked for the anthocyanins from the black soybean for antidiabetic properties. I have isolated a compound phenolic amide from the laba garlic. This laba garlic is a garlic which will be soaked in the vinegar, which is also one of the traditional Chinese medicine. So I have evaluated the antioxidant and cytotoxic properties of this compound against the liver cancer cell line and normal cell line. Other than that, I have isolated the Simiginosera compound from Simifiga genus, and these plants I have utilized for targeting the human breast cancer cell line. So the next project is I have prepared a hydrogel based on the nanocellulose from the garlic straw, and I regulate the release of allicin and its cytotoxicity. So I'm not going to explain all these things in detail. I'll just give how I isolated the compound and how I evaluated the biological properties by sharing two of my research project. Okay, the next slide. So this is my first project. So here first we have collected the bark material of the plant named Xanthosylum retsa, and then we soaked it in the methanol. So many types of organic solvents they will use to extract the compounds from the plants. So mostly we will utilize methanol or ethanol to extract the components metabolites. As I mentioned earlier, primary metabolites and secondary metabolites. So since we are targeting the secondary metabolites, we are using solvents such as methanol or ethanol. So here I used methanol. With the help of ultrasound acid extraction technique, I obtained a crude methanolic extract. So this extract will contain a diverse classes of compounds, not only a particular compound. So I want to know which compound is major responsible for the biological properties. So I subjected to liquid-liquid partitioning. Here I followed the traditional conventional process. So I followed use a separating funnel, and I separate the extract into fractions with the help of various solvents with increasing polarity. So based on the polarity, the compounds will separate. Okay, so I used the solvents called hexane, chloroform, ethyl acetate, and butanol. So here the hexane fraction will have non-polar compounds. And then after that, once I isolated the non-polar compounds such as fatty acids, or steroids, or everything, then I go for chloroform fraction. In this, I have isolated different class of compounds like alkaloids and other terpenoids, some things. Then once I isolated the chloroform fraction, then I go for the ethyl acetate fraction, and from the ethyl acetate fraction, we will obtain a flavonoids and phenolic compounds. Then finally, we use for the more polar compounds, we utilize butanol and water. So based on the polarity of the solvents, the samples will get separated. And once these samples get separated, we will further test for the biological properties to identify the most active fraction. Okay. So from this, we tested against the cancer cell line and normal cell line and other biological properties like UV protective properties and those things and all. So from this biological test, we identified which fraction is best. So that fraction we further collected, and this fraction is subjected to isolation process with the help of column chromatography, high performance liquid chromatography. So once by a separation technique, we isolated the compound, and that compound we will identify what compound is present inside the active fraction, which is responsible for this biological properties. So we will identify the compounds in the with the help of spectroscopic techniques like NMR, nuclear magnetic resonance spectroscopy, LC-MSMS, liquid chromatography, mass spectrophotometry, and we will identify the major components present in the fraction and the compounds. And once the active compound is identified, that compound is further tested against the cell lines, and we will target the biomarkers. So what are the biomarkers it's targeting and how it's targeting, whether it is expressing the biomarker or inhibiting the biomarker. Once it is identified, then the active compound will be have been selected, and that compound were subjected to a formulation. Nowadays we are using a sunscreen, right? So it will have many types of compounds which will be present inside the normal sunscreen, because that compounds have the biological properties such as it will absorb the UV light. So I have formulated a cream, sunscreen cream, and I evaluated the biological properties of the sunscreen. So from this project, I have published four papers. I managed to publish 4 papers. Next, so from this study, I identified that the ethyl acetate fraction of Xanthoxylum redsabag offers the significant SPF value, UVA UV absorption spectrum, good free radical scavenging property, anti-elastase and anti-collagenase property. Here, these anti-elastase and anti-collagenase properties are responsible for our skin tensile strength, because these two enzymes normally will break down the collagen and elastin present in our skin cell. So if these enzymes are highly expressed in our cells with the help of UV light, automatically it will damage the collagen and elastin present in our cells. So automatically what will happen means, once these protein called elastin and collagen get breakdown, our skin will get wrinkled. So now my plant extract have a capacity to target that enzyme. So that will inhibit that enzyme, and it will prevent the breakdown of active protein called elastin and collagen. So it will enhance the wound healing properties, as well as it will enhance the it will what is it enhance the glowing skin. Okay, so this plant extract has the that particular property. So moreover, this extraction is not this fraction is non-toxic to HGF cells, and it significantly prevent the UVB induced uh, biomarkers such as MMP1, MMP3. This is matrix metalloproteinases. So these markers will get inhibited with the help of this active fraction. So with the help of LCMS and GCMS, gas chromatographic and liquid chromatography, mass spectrophotometry, I identified 38 compounds and isolated 6 compounds. And we identified, at the end of this study, we identified the active compound. Hesperidin is one of the major active compounds. This is the lead component, which is responsible for the UV protection in this particular plant. Okay. So then I mentioned, like, the plant has been the active fraction has been subjected to formulation, and they managed to form a sunscreen lotion, and that sunscreen lotion also offer a good SPF value and moderate UV and UV ratio, and this cream act as a have a good pseudoplastic behavior, which is suitable for skincare and cosmeceutical products. So from the by the end of this project, I gave this conclusion: how this natural source can contribute to the medicinal application or cosmetic application. Next, this is my another project. I think I will just give a brief introduction about this. So same way, I have extracted the compounds from the fungus, and then I separated and tested for their oxidative stress in liver cell lines. Okay, but here the extra thing which I did is I have formulated this active fraction into fungisome, which is like a nanoparticle, because normally some components like phenolic and flavonoid compounds are polar compounds which cannot enter into the lipid bilayer. So the thing is, like, we need to pass the extract into the cells. How we can? So we have synthesized into a nanoparticle with the help of some other components such as silver or gold. Here I use phosphatidylcholine, which is a lipid, which is one of the major components present in our lipid bilayers, the cell membrane. So I encoded the nanoparticle, encapsulate the extract into a nanoparticle, and then I have tested their biological properties to enhance the drug delivery. Okay, this is another stage and which falls under the scope of pharmacognosy. So from this project, I managed to publish 2 papers. Next slide. So from this project, the thing is like among the different types of solvent, ethylene fraction possesses the good antioxidant and anti alpha amylase and alpha glucosase property. The formulation which I have did when compared to the normal extract, the formulation have a good bioavailability in the cell environment, and the both samples were non-toxic to the cells, which is now I tested on normal liver cells and cancer cell lines. Okay, next slide. So these are my current research projects where I am evaluating the photoprotective and anti-cancer properties of Moringa concanensis against the oxidative damage induced in LO2 cells and HB2 cells and normal liver cells. Next, I'm testing a nanoparticle. I'm synthesizing a nanoparticle from the peel waste. Nowadays, many fruit peels and fruit peels, vegetables peels are available in the environment, so which are throwing as a waste. So now I am planning to utilize those waste into a nanoparticle, those waste to a biological proper for a medicinal application. So I have collected the chickpea waste and I have synthesized it as a nanoparticle which has a good antimicrobial properties, anti-cytotoxic properties. So this will be further considered for any biological biomedical applications like wound healing, dressing, or active packaging. Then I'm working on the phytobiotic project Moringa oleifera. So we are targeting the gut microorganisms. The last project which I am working is like I am targeting the drug resistance, Taxol drug resistance in non-small cell lung cancer with the help of active component present in the garlic, allicin. So I am targeting the allicin in reversing the Taxol resistance in cancer cell lines. These are my current projects. Next. So far I have published 37 publications and I filed one patent for the photoprotective properties, and I have copied one fabric which is like innovative germ-free fabrics. In this fabric, I have coated a nanoparticle which contains pineapple peel extract, and that peel extract nanoparticle are coated in the cloth or fabrics to focus like antimicrobial clothing. Okay, and I have published one book chapter in the field of edible mushroom and their health effects. Next. So these are my selected publications, which are more related to pharmacognosy as well as functional food applications. Next. So what are the challenges which I have faced and normally the world is facing in this natural drug discovery process? The first thing is seasonal and environmental variation. Normally, we all know like there are like every time and every year there are many different changes in the season, and the environment also changes because of many pollution. So because of this, there might be changes in the metabolic composition. So this will affect the natural drug discovery process. Next, the solubility and stability of the sample. Normally, if it is in a plant extract, if it is in a plant, the compound will be stable. Once we isolate it, sometimes their stability will get lost, or else sometimes it will have soluble issues. So that will also be a challenges in our natural drug discovery process. And complexity of the mixture after fraction. So I already mentioned like we will isolate the extract and we'll go for fractionation, but sometimes the fraction will contain different types of mixture, which is very hard to isolate, hard to separate. Okay, after that, once we isolate it, we will get a very less amount of samples. So that is like another drawback. We will work for almost like 6 to 7 or 8 months, but we will get a very limited amount which we cannot further process for any biological process. That is a major challenge which we face in natural drug discovery process, and the bioavailability. So once the compounds have a good biological property and everything, the thing is like once we are taking into the market or taking into treating with the animals or anything, the bioavailability of the sample or active compounds will be very less. So which we cannot achieve the goal. That will be also. Moreover, the time consumption and cost, lack of regulatory framework, all these are the challenges in natural drug discovery process at this moment. Next. So the suggestion is like adapting the innovating approaches in pharmacognosy, such as nanotechnology, bioinformatics, molecular docking, and omics approach, like proteomics, genomics, and metabolomics approach could be most advantageous in overcoming these constraints. So if we adapt this nanotechnology or proteomics and genomics approach, I think we can fight against the challenges which we are facing in the natural drug discovery process. Next. So these are all my collaborators which are still supporting for my research field from China and Malaysia as well as from India. So these are all the collaborators. Next. So this is my research interest group in University of Malaysia Terengganu. So these are all the lecturers who are involving in biological science program. So here we are working on three research domain. First one is health research and disease control. Second one is based on the environment where they are working on conservation and environmental sustainability. And another domain is on food, which they are working on the food security and quality development. So in case if you in future if you guys need to contact us, please feel free to contact for any collaboration or any further studies. Okay, that's all. Next. Thank you.
Okay, thank you so much, Dr. Ramesh. That was very insightful. Okay, so students, I think we can take some time for questions. Do you have any? You can drop your questions on the chat box. Okay, if you, Dr. Ramesh, you don't mind, can I ask you from
Yeah,
my
yeah,
side?
yeah. Please,
Okay.
please.
Okay, you were saying about the primary and secondary metabolites, am I
Yes.
right? So you say that we chose usually for studies for medicinal therapeutic application, we always choose secondary metabolites, not
Uh,
primary.
Not always. We are major part we will choose secondary metabolites, but nowadays we are targeting the primary metabolites as well. But
Why is
like
significantly secondary metabolites are being chosen for therapeutic application?
Okay. So in primary, for example, secondary metabolites will be for extrinsic function. So it won't affect the primary source, the main organisms. So if you're targeting for the, what to say, primary metabolites, the source of primary metabolites will also be less because it will be already consumed by the plant or the
Oh,
host. But the secondary metabolites will be like a backup. So if we target that, it won't affect the host as well. So it is like mutual benefit.
okay, understand. Okay, and then the part where you were saying about the identification. Okay,
Yeah.
if just say in a plant that we found after we do the screening, and then we identify that there is such compound like maybe perhaps tocopherol. Okay, they're
Okay.
responsible for anti-inflammation or and so on. What is the instrument that we use to basically physically isolate them?
So there are many techniques are available. Like we can use thin layer chromatography. Chromatographic
Thin.
techniques. I mean, there are many techniques like chromatographic techniques. You can isolate the compounds. Okay,
We
chromatographic.
can, isn't that to identify the compound, how to physically, if let's say this now I have this fraction,
Okay,
okay, and then I know I've already run the chromatography technique, and then
okay,
okay, now I know this is my target compound. Now I need
okay.
to physically remove that from my fraction and have this pure compound in my study to further. So what instrument we need to use?
HPLC. HPLC or
HPLC.
column chromatography.
Okay, okay, understand.
Because you can, if you identify the compound, then you can run a standard. You can buy a standard, and then you can first identify which peak is your peak, particular peak in HPLC. Then you can use a preparative column chromatography, and then you can isolate the compound. That will be the
Okay.
most easiest option.
Yes.
Or else you
So
have to go for the column chromatography, and you have to wait for a long time. And we don't, if you isolate at the end, you don't know which compound is that. But if you
mhm.
use HPLC, then it will be easy to identify the compound. If it is a
Okay.
known compound. If it is unknown compound, then we have to go for another technique. Like we have to use NMR or LCMS-MS to identify the fraction. I mean the fragments, and then we have to identify a new compound,
Okay.
which involves like chemist. most
Hmm.
natural product chemists can support us.
Okay. So, and then when you do this comparison
Yeah.
between the traditional and also the modern technique, I see that I noticed that traditional is more of a downstream. That means you start from the compound and then going to your target
Yes.
disease.
Yeah.
Whereas in the modern technique, it's about upstream, then you're going down to the downstream.
Yeah.
Is that significant difference between the traditional and also modern?
Yes. Yes. The basic difference is the main difference is that is the thing. So in
Okay.
this, like if you go in traditional medicine, we don't know the target. You will just collect the sample, but based on the traditional, literature review or something, literatures or any traditional information, like this plant has been used for diabetic, this plant has been used for wound healing. We will on that word of mouth, we will take the select the plant, then we will go for the target. But in modern medicine, we will identify the target, which biomarker we are going to target. Then we
Mm.
will focus on it.
Do you think there is any pros and cons in between this
Of course,
method?
there are in both techniques we have a pros and cons.
Mm.
But
Which one do you think is better, traditional or
based
modern?
on my experience, both have a equal weightage.
Okay.
Uh, but if you are starting from the scratch, then I think you can start from the traditional approach. Then you can adapt the modern method to advance your research.
Okay.
It's based on
Okay.
my
Sorry, I'm going to your personal research where you were talking about UVA and UVB, right?
yes,
So, as you mentioned, the wavelength difference between these three rays, okay, is longer and shorter. Am I right? So
yes,
UVC, we don't have to worry about UVC at the moment,
yes,
okay, because they are very short, so they cannot penetrate through our atmosphere. Am I right?
yeah. Because of the ozone layer, it is prevented, and then it won't reach us. It won't affect
Okay.
us normally.
So, if UVB and UVA is our concern,
Okay.
definitely because of the length itself, it
Hmm.
has different pathological impact in our body,
Yeah,
isn't it? So, what
yeah.
is the difference between UVA and UVB?
Normally, UVB will target only the epidermis in our skin layer. It cannot penetrate
UV.
into until the dermal layer. But UVA will penetrate into the glass, and it can easily penetrate until the dermis.
Okay. So, the difference is if let's say we're talking about UVB, it's usually the one causing the burns
Yes,
because
sunburn.
it's superficial.
Yeah,
Okay. So our sunscreen, the commercial products, they usually target UVB or UVA mainly
yeah.
because uh, yeah.
Yes, initially they will targeted the UVB. Actually, the in commercial market you will get the sunscreen, right? That will have
Yeah.
SPF value. If
Mhm.
they provide only the SPF value, then it is mainly for UVB protection, because
Okay.
the SPF value will be calculated based on the UVB range only. Okay,
Hmm.
but if your sunscreen product has like it has a boot star rating or any UVA and UVB ratio, then it will target the uh, UVA and UVB as both. So
So that
nowadays, most of the sunscreen product they will have UVA and UVB ratio product.
does that this SPF 30 and 50, is that
Yeah,
what you're
the SPF
saying?
30 and it is the like percentage of absorption.
Okay.
How percentage of UVB absorption, not UVA absorption. The SPF value only reveals the UVB absorption. So if
Oh,
you
okay.
want, if you are looking for more product, a more efficient product, you have to search for SPF as well as both UV and UVB ratio, or else some booster rate.
So the 30 and 50, it's not related to the race that we are talking about now.
No, only on the percentage of UVB protection, percentage of only the UVB
Absorb-
protection. Yeah,
Oh, okay, understand. Okay, and then the last question from my side, sorry.
yeah, yeah, no
Okay,
problem.
so you were saying about the silver nanoparticles, am I right, injected to enhance the absorption by the cell. So if we are using such particle, what is the chances of our body rejecting these particles?
That's what initially we will test on the normal cell lines, okay, based on the concentration. So if there is we will check for the different ranges of concentration, okay.
Hmm.
So we will check for the cytotoxicity as well. Sorry, can you ask again? So based on
Uh,
the cytotoxic effect and the bioavailability, we will check for the in vitro drug release and the concentration of the drug inside the environment, inside the cellular environment. Then we can identify whether it is rejected or it can easily adapt.
mm. So there is chance, but we have to do certain what do you call screening or certain evaluation to get the optimum
Yes,
concentration.
bioavailability we will check. We will check the in vitro drug release in the in vitro environment. Then we will go for the clinical environment as well.
Okay, thank you. Thank you so much.
Okay.
Appreciate you being here. Okay, Dr. Ramesh, thank you.
Okay.
I think that's all for today. I hope
Okay.
we can have you again here.
Yeah, sure.
So we will see you next time around. Thank you.
Thank you so much. Bye.
Okay.
Thank you
Bye.
all.